This method can help answer key questions in the field of exercise biology, such as how does exercise response differ for animals of different ages, and what genetic pathways control exercise response. The main advantage of this technique is that it allows researchers for the first time to measure activity in flies performing rotationally induced exercise in real time. Demonstrating the procedure will be Louis Patrick Watanabe, a graduate student from my laboratory.
Prior to the planned experiment, set up crosses of male and female flies so that age-controlled progeny can be collected for the experiment. To begin, load 8 glass specimen tubes with 10 anesthetized test flies per vial. A standard experimental group consists of 10 vials of 10 flies of each sex. To load the vials into the REQS, insert the vial into a slot of the activity monitor from the front. Then, put the second cap on the vial and center the vial, moving the O-ring as needed. House the REQS in an incubator for controlled environmental conditions. Allow the animals to recover from the anesthesia for an hour in this incubator before proceeding.
For assays longer than 8 hours, vials containing food that is very securely attached to one end of the tube may be used.
Following the acclimatization period, power up the device to start the rotations. Adjust the speed to the desired speed, such as 4 RPM. No tubes should make contact with the bottom surface if they are properly positioned.